was, let me answer your question since you're asking it, because I will answer this very clearly. When was there recording when you were taught in a classroom? Did you not take notes? Are you not supposed to take notes? Why should you relax with your hands in your pockets and listen and go back to the lecture? Are you not supposed to take notes and listen? Let us have a clear answer to this question. Come on. 30 years you have been taught, we have been having academics, I have been teaching. Never was there a online teaching and never was there any recording of the lecture notes or the lecture which was given by the teacher, and students were supposed to take notes of all the important points for the revision. So Mr. Achinta, can you answer that question?
Sir, but notes is okay, but like if somebody is not able to make notes, then what he should do?
Why will you not be able to make notes? Can you tell me why? Give me
Like
one reason why you don't have a
everything is
paper
not
and a
clear
pen.
at one go. Like it takes some time to understand the concepts.
What happens? What happens when then you have to ask questions if it is not clear?
So, but classroom teaching is really different from the online teaching also. So,
Absolutely right. But how does that justify recording, which actually sort of also ensures that 80% of you students don't attend the class? And because it is being used as a justification for not attending the classes. And then when performance is poor, then, who's to blame? This recording business has actually ensured non-attendance and has also actually ensured very poor understanding because you're not interacting in the class. You're not asking for doubts to be clarified. You are a passive spectator rather than an active participant. And that is something which is very, very serious and is going to cost you very heavily in the long run. This whole reliance on the fact that there will be a lecture which is anyway recorded, and anyway the questions that will be asked will be part of the lecture, and I have to somehow search out from the lecture which part of the lecture is the question that I have been asked for, is a completely erroneous assumption. You have to be active when you want to learn. Passive learning is the worst form of learning ever. Whatever. I think it's a tragedy which, as young kids, you are not understanding what is happening here and what is going wrong here. And well, we teachers have to blame, but it is important, at least for me, to point out that your perspective has changed for the worse. And it's very, very dangerous. Anyway, the story that I was going to begin with. So I gave you this problem, and it's a real problem. Fructose has been produced in very large quantities by industry. Do you know why? Can any of you tell me why fructose is a very popular product in the industry? Hello? We need interaction. Just now, that's what I was talking about.
So it can be converted into other simple sugars.
Sorry?
Maybe because it can be converted easily into other simple sugars.
No. It is a simple sugar. It is actually produced from, let me tell you, what is sucrose? What is sucrose? Sugar, चीनी जो आप खाते हैं. ये क्या है पता है आपको? What is sucrose? It's a disaccharide. Hello? क्या है sucrose भाई? What are the two simple sugars that make up sucrose?
Glucose and fructose.
Glucose and fructose. Glucose plus fructose. Now, if you heat it in acid conditions, you will split this bond. I hope you know what this bond is. I've been asking so, and you will get glucose and fructose. Why is it better to split it before adding it into as a sweetening agent? Can you tell me? And then, of course, you can also use isomerase, because these are isomers. You can use the fructose isomerase or glucose isomerase, which are actually the same thing. Whether it's glucose isomerase or fructose isomerase, it actually means the same enzyme, fructose isomerase and glucose isomerase, to convert glucose to fructose. But remember that the delta G is about 0 at say 60 degrees. It is remember delta G is delta G naught minus Tg delta S. So at different temperatures, the delta G is different. But at the temperature it is converted 60 degrees centigrade, this is about 0, which means that K equilibrium is 1:1. Right, so that's a separate point. But why do you want to split this into glucose and fructose? Can you tell me what is the advantage that the industry gains out of it? Because fructose is 10 times sweeter. Fructose is 10 times sweeter. And because it is 10 times sweeter, you have to add less in Coke when you split this. So they never add sucrose. They take sugar, they acidify it, and they heat it, and acidification and heating is enough. Of course, there are enzymes which also break down glucose and fructose, sucrose to glucose and fructose, but instead of a cheaper method would be to acidify and heat, and it will break down. And that ensures that the sweetness of the sugar becomes much higher. Glucose is less sweet than sucrose. Fructose is 10 times more sweet than sucrose. So the splitting of the sucrose into glucose plus fructose makes it actually about 5 times sweeter, because you get 50-50 of glucose and fructose. Now people then realize that it actually makes sense to take this glucose, separate glucose and fructose using methods which earlier were very difficult, but nowadays with the coming of something called simulated moving bed chromatography. Have you heard this word, simulated moving bed chromatography, SMB? Any of you? No. Yes? Hello? So when I say words, I suppose if somebody should object and say, yes, we know heard about it, we have not heard about it. आपने सुना है आप लोगों ने? Simulated moving bed is basically the fact that very briefly, it is a much more efficient way of separating molecules like glucose and fructose, because when you separate in chromatography columns, you have to add a pulse and then get a glucose pulse and then fructose pulse. So this is a non-continuous operation. But simulated moving bed is when the bed moves backwards in a countercurrent way. Actually, a bed cannot move because it is made up of solid particles. So solid bed cannot move, but it moves backwards, this moves forwards. You simulate the movement of backwards by clever methods. And that is why it's called simulated moving bed. The bed is being simulated as moving. So you have a countercurrent method, and you can supply fructose and glucose simultaneously, and you can get fructose on one side and glucose on the other. So it's a continuous system. It's a continuous chromatographic system, and it is much more efficient. Someday, if I get a chance, I will teach it. I taught, I think, your senior batches this subject because of some time, and you get actually fructose. And now the glucose that you get, people have now started converting it to fructose by the isomerase enzyme. And again, that fructose that they get, they also sort of use for this. So, and then the remaining glucose is recycled back, and so you get 100% conversion by first taking out 50%, the glucose is fed back, again digested, and finally you end up getting a fructose solution. Remember, I mean, it's easy enough, right? I mean, you have glucose coming in, what we are going out is glucose plus fructose, and the fructose goes out, and the glucose sort of is recycled back into the system. So you have a recycled stream, you have glucose stream here, and fructose coming out here. And so obviously, the whole purpose of this conversion and this money is spent only because of this very rather childish thing of this being 10 times sweeter. And more plants were built up to make, and earlier it used to be called high fructose corn syrup. And this is the way it used to be sold: corn syrup, starch, essentially glucose, starch broken down to glucose, was converted into fructose. So it was high fructose corn syrup, and this was sweeter than normal corn syrup because glucose is not very sweet. And this used to be added to Coke, and that is what makes it so sweet and nice, of course, other than carbonate. All kinds of harm, but then Americans tend to drink Coke like we drink water. Okay, so, and of course pastries and many things. And then a lot of fructose was made, and then at some point of time, it was discovered that fructose consumed in large quantities can cause liver damage. And also there is a there is an epidemic of diabetes, and that is true much more for Indians than for foreigners because we don't measure it, and overweight, obese people, and lots of heart troubles. And sugar is now considered the biggest killer in this world, much more than the war in Ukraine and any other. All the wars put together, all the gunshot wounds put together, all the knife wounds put together, all the muggings put together, kill less people than sugar does every year. That is a major victory for technology in the 21st century. Sugar kills more people than people who die of violence. So therefore, this becomes a serious problem, and because but it kills indirectly, so it's not obvious. Now, if you convert sucrose to psychose, this is an amazing sugar. This sugar does not get absorbed by the body. There are no enzymes to degrade it, and therefore it adds no calories. So every health conscious person, every diabetic person who is craving for sugar, for a cake, for rasgulla, for chocolates, for Coke, can now use psychose. But psychose is a very difficult thing to make because of fructose to ficose. So one industry was making it, and I was a consultant, and so that's why I asked this question. Now it turns out that I told them, please scale up, because this is an epimer. So I told them, please, it's called fructose epimerase or psychose epimerase. These are epimers of sugar, isomers of sugar, that depends on the COH bonds and so on and so forth, that chemistry. So this epimerase, and the conversion is 25%. So again, you need an SMB to separate the fructose and psychose. But epimerase enzyme converts this. So I told them they have cloned it in E. coli, produce it, and so we coming to the immobilization now. The story is getting complete. So when we have the epimerase, I told them, please scale it up. You have produced it in an E. coli in a flask, and then they scale it up. So when I went there, they had taken a bottle and they had put fructose. They had taken a small flask, put fructose, and then left it for some time, added the enzyme, and after some time they got their psychose, or rather 25% psychose and 75% fructose. Now it turns out, I said, scale it up. So when I went next time, they had taken a 5-meter cube tank and added the enzyme. I said, this is not a scale up, this is growing at a large scale. How do you scale it up? They said, well, we have made a lot of fructose. But then I asked their management, how much fructose do you want to make? Psychose do you want to make? They said, we will set up a small plant in the beginning and make it larger later on. I said, what do you mean by small plant? They said 100 tons per day of psychose. And remember, this is a 5% solution or 10% solution. So if the fructose here is a 50% solution, let us say very thick concentrated 50% solution, the psychose that you will get is a 12.5% solution. And 37.5% of will be the equilibrium. So 12.5% concentration. So to make 100 tons per day, you think of the size of the tank that you need to use. So I started laughing and I got very angry with them. I said, this is not a scale up. Where are you from? And none of them were engineers. And so we come to the main point. I said, you cannot do this. Because how do you convert? You convert by reusing the enzyme. And to reuse the enzyme, you have to have a column. And you have to bind the enzyme to a matrix. And then all the issues of mass transfer, etc. come in. But really, we will not talk about that. What we will talk about is how do you bind? How do you bind the enzyme to a matrix? And that is what Kshitij will explain to you. This binding of an enzyme to a matrix obviously allows you to reuse the enzyme again and again and again, because once you have bound it, this column sits there, the enzyme sits there and converts your fructose to psychose. Of course, not fully, but A to B, without any trouble whatsoever, once you have set up the design. And therefore, and that is why I said about every subject in the class, and I say this for this class as well, designing immobilization strategies, forget designing immobilization strategies, designing matrices that will carry enzymes is the whole field in itself in biotechnology. One of the richest people are people who design matrices for enzymes. Because the ability of the matrix to trap enzyme, to trap it in large quantities, much larger quantities, to trap it such that it does not degrade or denature or change shape, so that its Kcat is the correct word to use. Kcat values and the Km values do not change. Affinity does not change while it is bound, because after all, when it is bound, the enzyme is, suppose this is where it is bound in the matrix. This is the part which is being used by the substrate. The ability of the substrate to enter and the ability of the product to leave is impacted by this matrix which is holding the enzyme. If it is holding it well such that this is not impacted, then neither Kcat nor Km are affected. But if you are holding it like this somewhere, then obviously this shape will change here in ways such that the binding efficiencies and the conversion efficiencies will fall. So matrix design is an art and practiced by the best chemists in the world, because it is a very, very profitable solution. Billions of dollars. So, we are not, that is the whole problem with this course. It actually only touches upon or introduces you to subjects which are of tremendous importance in themselves, but at least you are aware of the fact that you do not know this and this is a subject that exists. And that is my purpose here, to make you aware of the fact that there are areas and fields which are exceptionally important. How much will it bind? How much will Km-Cat change? And the most important of all, how much will the stability get changed? To give you an example, we tried out hundreds of matrices. And then after binding, there was 5%, 10% loss in the best matrices. But the most important point is this enzyme now becomes stable for 2 months. And when I tell them it is becoming stable for 2 months, they laugh. They said, we have enzymes which are stable for 6 to 8 months. Notice that enzyme sits here for 6 months with fully operational ability converting the enzyme. So you see the fantastic advantage of immobilization. 6 months it is going to sit there. Obviously in 6 months it will degrade, but it degrades slowly. It will lose its ability slowly. Now let me ask you. Why not ask you? There is something called the turnover number. Have you heard of this thing? Did I teach you this? Yes, no? Hello?
So we heard of it.
You heard of it? What is the turnover number for a typical enzyme? Choose some Kcat, choose some Km. Turnover number is the number of substrate molecules that one enzyme molecule is able to catch and convert to product per second. What is this number typically? Is it 1 per second? One enzyme molecule, one molecule once per second? It is a time constant, right? In a sense, it is the inverse of a time constant. How many molecules of substrate does one enzyme molecule convert per second into number of molecules of substrate? That really makes sense. Of course, please remember that this molecular weight is much larger and this molecular weight is much smaller. So fructose epimerase may be 50,000 Daltons, or 50,000 is the molecular weight, and sugar and this is 180. Molecular weight is 180. So obviously, but the turnover numbers go from 100 to a lakh or higher. And imagine you have a turnover number of even 1,000, forget about that, per second, and you are running it for 6 months. So look at the number of molecules each molecule of enzyme converts, and it is greater than 1 billion. And that is the advantage of immobilization. And therefore, when I actually calculated that we had to make 100 tons per day of fructose, and they started telling me what about the enzyme plant, because the enzyme has to be GMP regulation, and let me not get into GMP and all that. I discovered that 100 liter fermenter, 200 liter fermenters is good enough for 100 tons per day of product. You are producing the protein in grams and you are producing tons. Because you're converting more than 1 billion molecules. So even if the molecular weight is 100 times larger, you are making 10 million grams out of 1 gram of protein. So you see the advantage of immobilization. So it is to do with stability, it has to do with non-changing KMK cat, it has to do with enzyme loading because then you can reduce the volume. I mean, this 24 hours in the tank, you know, finally in the reactor with appropriate modification of mass transfer effects, we could convert the substrate into product in 10 minutes flat. 10 minutes was the residence time of this reactor. Because this is heavily loaded with enzyme. So these are the wonderful properties of matrices and how you immobilize. Now we will not get into matrices. Much of it is actually beyond our scope, also because industry holds these things very, very carefully close to its chest, because matrices sell for millions of dollars precisely because they do this, what I'm talking about. But immobilization and designing matrices is a very important, because each matrix is actually tailor-made, designed for a particular enzyme. Can you think of how you can tailor-make a matrix to design for a particular enzyme? Can you give me an idea? Can you think of a mechanism by which I could tailor-make matrices for a specific enzyme A, and another one for B, and another one for C? Can you think of something? Guesses from your understanding of biology? हाँ भाई, कुछ तो बोलिए आप लोग. Make a guess.
So probably first we have to like select the material of the matrix based on what our enzyme is.
नहीं, बट मटेरियल तो आपको बाद में सोचना है ना. आपको बेसिकली उस एंजाइम को बहुत बढ़िया तरह से बाइंड कराना है. And it is a designer, of course, one of the methods that we are talking about. I am talking about biological, easy biological solution. How do you find out what captures a particular enzyme, which is a protein, the best? What are the things that capture proteins the best? क्योंकि मैट्रिक्स आप चेंज नहीं कर पाएंगे ना. मैट्रिक्स अगर इनऑर्गेनिक सिलिका है, मान लीजिए थोड़ी देर के लिए, सिलिका इज अ वेरी गुड मैट्रिक्स. Sand, okay, silica. Now you have silica matrix. So let me sort of rub it out and say. So I have silica. And really these matrices are made like this. So these matrices are like this, that there is a core matrix. And then there is something like a capture, which you attach. And this is specifically. And this capture then picks up the matrix of your choice and captures the enzyme and holds it like this. And this is the enzyme working at. मैंने ये drawing बनाया आप लोगों को hint देने के लिए. See, each of these is holding an enzyme. And these enzymes are then converting. So this is the base, this is the specific thing that the matrix has, and this is your enzyme. What is the blue thing? Can anyone, if you make a guess?
The antibody.
Obviously. तो ये enzyme लीजिए, fantastic answer. Enzyme लीजिए, उसका antibody रेस करिए, उसको किसी चूहे हुए में पंच मार दीजिए. अब ये सब तो हम लोग को बायोलॉजी तो आता नहीं है, इसलिए मैं चूहे में पंच मारने की बात कर रहा हूँ, है ना. बट चूहे या घोड़े या मतलब गिनी पिग या I do not know, whatever you punch it in, raise antibodies and see whether you can sort of attach these antibodies to the silica matrix, and then the enzyme will become an attachment to this. Obviously the antibody will have to be then attached with something that you have some chemistry for, and then the enzyme will, but when it attaches, you have to make sure that this attachment is not at the active site of the enzyme. As I said, if this is the active site, and it should attach at this point. So आप इस point का antibody raise करिए, so that ये pocket खराब ना हो, and all kinds of clever tricks. So immobilization is a very huge subject, and we will introduce it to you in a very simple way. And we have Kshitij with us, who will take over now. So I just wanted to tell you the importance of the subject. I wanted to give you an introduction about the power of the subject and how useful it can be. It can change the world. You understand? You have good matrices producing enzymes, even very expensive enzymes. You know how much it cost us to make 1 kg of fructose, of psychose? The management had calculated that the enzymatic cost of conversion of 1 kg of fructose to 1 kg of sucrose, the enzymatic cost only. I'm not talking of water and manpower and SMB, etc., etc. 1 kg of sucrose, they had calculated that this cost was rupees 25. And when he called me over, he said, please bring it down to rupees 10. Because this is important, otherwise there's too much of cost because I cannot sell it. So the enzymatic part of the proportion of the cost of converting fructose to sucrose per kilogram was rupees 10 per kg. What they wanted rupees 10 per kg, and it was costing them in initial calculation as per their molecular biology effort was 25. With immobilization, it came down to 15 paisa per kg because it was solid for 2 months. There's hardly any enzyme cost. But the enzyme cost did not go down. What went down was the fact that it was sitting there for 2 months and converting. So it converted billions of molecules. So from 25 to 10 rupees, I told them it's not 10 rupees, it is just 15 paisa. मेरी तनख्वाह नहीं बढ़ाई गई, वो अलग कहानी है, आप लोगों को बाद में बताऊंगा. So anyway, so Kshitij, you are there? You are here?
Yes, sir. I can.
Yeah. So let us now. I will give you sharing. So I think now all of you can listen to the next part of the immobilization. All of you okay? Any questions that you want to ask? So I stop sharing.
None currently, sir.
And where is it? Okay. कैसे होता है? More actions. Okay. Meeting options. ये तो नहीं हो रहा है. Kshitij, how do you want to?
Sir, maybe if you can see my name on the screen, if you move your pointer over to my name, there's more options. There's 3 dots that come up.
Oh, well, I can send a message to you, but. Okay.
Just a moment, sir. I'll look it up once.
Let me see. I don't know whether I can do this. I have to sort of check out. No. I think we will forget it this time. All of you kindly do not.
Sir, I think I found it.
Sorry.
So do you see a participants list?
I can see make a participants list. Yeah, I can get it. Yeah.
Yes, sir. So do you see my name under attendees over there?
Okay, let me roll down and see. Yeah.
So if there are 3 dots there should be a make a presenter option.
Okay. So I made you a presenter now.
Yes, sir. That's perfect. I can now share my screen.
Okay, so you share your screen. All of you guys can hear me and Kshitij talking.
Yes, sir.
Okay.
Yes, sir.
So I will stop here and let Kshitij show you some of the slides of the basic techniques of immobilization. Show you a experiment which should ideally should have been taken care of, and then go on to the next step. Okay.
Sure, sir.
Kshitij, can you start presenting?
Yes, sir. I'll begin right now.
And you can show your own introduction, and I will be leaving shortly, but you can always give me a call on phone if you need me.
Definitely, sir. Yes.
You start the presentation and I will leave, unless you people have some questions.
Okay, so can everyone see my screen?
Yes.
Yes.
Yeah.
All right, perfect. So, hi everyone. My name is Kshitij. I'm a 2017 entry student. I'm in my 5th year right now, and I'm going to be giving you a basic introduction to enzyme immobilization. So, yeah. So, if anyone has any doubts in the middle, you can always interrupt me. You can always stop me right there. I'm going to be putting on the slideshow right now. So, here we go. Just a moment. Yes. So, we're going to be talking about enzyme immobilization today, a bunch of techniques, what the advantages and disadvantages are, and so on. So the learning objectives we have are to basically understand the immobilization of enzymes or whole cells, what the difference is between both of them, of course, what advantages and disadvantages we have for immobilization, what are the applications of it, the support matrices that we use in these techniques, what the various methods are of immobilization. There are basically 5 which we are going to explore in this presentation.
Shilj, I have one doubt. Can you explain what is immobilization?
Yes, we're coming to that, Achintya. I'm just telling you what we are going through in this presentation. I'll come to that, don't worry. Then we're going to move on to immobilization of whole cells. Afterwards, we're going to talk about the advantages and disadvantages of whole cell immobilization and the difference between enzyme immobilization and whole cell. And finally, we're going to conclude with the methods of whole cell immobilization. So the first thing we're going to cover is what Achintya just asked. What is immobilization? So it's basically defined as the imprisonment of a cell or an enzyme in a support matrix. So what basically happens is, like Professor Mukherjee just said, the enzymes which we use in most industrial applications or even in the lab, they're generally very expensive. So let's say you have a substrate that you want converted to a product and you want to speed it up using an enzyme. And you can add the enzyme to it directly. Let's say the enzyme is a protein, it's in a liquid form, for example, and you put it with the rest of the reaction mixture and you're trying to convert your substrate to a product. Now, the problem with that is once you add the enzyme to it, it's very difficult to recover that enzyme. It might also contaminate the product. There might be a lot of problems. So what we basically do is we try to bring in an aspect of reusability via immobilization. So what we do is there's a support matrix inside which we sort of imprison the enzyme so that, for example, if we have a single bead in which the enzyme is immobilized, and if we add that bead to the substrate reaction mixture, the substrate is going to get converted because, like you all know, enzymes are not used up in the reaction. They just act as catalysts and they just speed up the reaction. So if we have it trapped in a bead, for example, we can just take that bead out and the substrate has already been converted to the product and we can reuse that enzyme again. So that's the basic idea of immobilization. The support matrix should obviously allow for an exchange of medium. Just a moment. Yes, there's a laser pointer. So it should allow for the exchange of medium. The medium contains the substrate or effector or inhibitor molecules, whichever, depending on the enzymatic reaction. This is a bit of history. The first ever immobilization technology was amino acylase by this organism for the production of L-amino acids in Japan. Now, 2 main advantages of immobilization are the first one is increased functional efficiency. The second is enhanced reproducibility. Moving on, what are the advantages of immobilized enzymes? The first one, which I've already mentioned, reuse, which is the most important one. Then as we move on, there's continuous use, there's less labor input, there's saving in capital costs, like the example you just heard Professor Mukherjee tell you about, from the rupees 25 per kg to the 15 paise per kg reduction. That's a significant amount. Then there's minimum reaction time. There's less chance of contamination of the products. The enzyme is more stable. There's more process control, and you have a high enzyme substrate ratio. Now, what are some disadvantages of enzyme immobilization? The uses in industrial applications are a bit limited, definitely. Loss of catalytic properties in some enzymes, because what you have to understand is when you're trapping the enzyme inside a matrix, for example. Now, sometimes it can be physical phenomena, sometimes there are actual covalent bonds that are used to trap the enzyme. So when you have covalent bonds forming between two species, it's possible that the enzyme might undergo some chemical changes, and because of that, it no longer has the property of a catalyst. So that could be one of the disadvantages. Some enzymes,
Actually, these are the disadvantages of immobilization of enzymes.
yes,
So,
yes.
like how immobilization causes loss of catalytic activity properties.
Like I just explained, Achintya. So what happens is immobilization. A lot of times they're just physical cross-linking, but sometimes they're covalent bonds that are formed between the enzyme and the matrix. So because of those covalent bonds, there might be changes in the structure of the enzymes. So I think you've all heard about the lock and key mechanism and about how there are active sites on the enzymes. So if the enzyme undergoes a chemical reaction, it's possible that its properties might change and it loses the catalytic property. Moving on, some enzymes, they become unstable. Some enzymes are inactivated by the heat generated, and there are very high costs for isolation, purification, and the recovery of active enzymes. Moving on, what are some applications of enzyme immobilization? In industrial production, we can use them for antibiotics, for beverages, for amino acids. In biomedical applications, treatment, diagnosis, and drug delivery. You can go through this list yourself. I'll try to make sure that the PPT is shared so that you guys can go through this PPT yourself as well. And the report that you're going to be submitting for this lab practical, it's going to involve a summary of this entire PPT. So I would suggest you go through it once yourself as well. So
Shishir,
basically,
can you go to the advantages of enzyme immobilization? Advantages of enzyme
yes,
immobilization.
Achintya, I know you want to take notes and everything. That's why I'll have this uploaded and you can go through it by yourself. Okay, we need to complete this by 4 and there are a lot of slides. So I'm moving on right now. I'll have the PPT uploaded. Don't worry.
Okay, okay.
So these are some of the applications of enzyme immobilization. Moving on, the techniques that we use, what are the various supports or matrices that we can use? The matrix is basically there to hold the enzyme. Now, what are some factors that we need to consider? It should be cheap, it should be easily available, but more importantly, the reaction of the matrix with the medium and the enzyme should be as minimal as possible. Because of course, you don't want well, you're trying to immobilize an enzyme for enhanced reusability. And what happens is when you put that matrix inside the substrate, the matrix starts reacting with the substrate. That is not something you want. So the reaction with the medium and the reaction with the enzyme should be as minimal as possible. Now, a wide range of matrices are used in immobilization of enzymes or whole cells, and these can be grouped into 3 major categories. They're pretty simple. There's natural polymers, synthetic polymers, and inorganic materials. So these are the kinds of matrices we use. Now, this is a list of some of the examples of natural polymers. You have alginate, you have chitin, you have collagen, there's gelatin, there's cellulose. There's a whole bunch of natural polymers you can use as a support matrix. Moving on, these are some of the examples of synthetic polymers that we use as matrices. They're generally ion exchange resins or polymers. They are insoluble supports. They have porous surfaces because you obviously need porous surfaces for an exchange between the substrate and between the matrix and the medium, because you want the substrate particles to permeate into the matrix and interact with the enzyme. So that's why you need porous surfaces. The porous surfaces trap and hold the enzymes or the cells. Some examples of these synthetic polymers are polyvinyl chloride, PVC. There's PEG, there's DEAE. I'm not really sure what the full form is right now, but yeah, I mean, these are just examples of the synthetic polymers. Moving on to inorganic materials, you can use zeolites, you can use ceramics, you can use silica, there's activated carbon, there's charcoal. There's a lot of examples for inorganic materials as well. So I'm going to pause here for a second and I'm going to ask, does anyone have any questions so far? Everything I've said so far, I mean, it's getting through to you guys, right?
Kshitij,
Yeah.
I just wanted to ask, like, do you have any figure which can explain the mechanism of immobilization clearly?
I don't think there's any, there might be some figures in the PPT as we go ahead, but not right now. Let me check just a moment. Something that might help explain it better. Yes. Yes, this is the entrapment technique. So you have the matrix over here. You have enzymes trapped in the middle of the matrix. You have huge pores where the substrate can come and interact with the enzyme. So this is the basic idea. Does anyone else have any doubts? So it's all clear so far?
Yeah, it's clear. You can proceed.
Okay, perfect. Moving on then. Yes, so now we're going to the methods of immobilization. There are 5 basic processes. There's adsorption, there's covalent bonding, there's entrapment, which I just showed you the picture of. There's copolymerization, and there's encapsulation. So we'll visit all of these methods one by one and explore them. So this is a flowchart of all the various types of um, techniques that you have. Moving on. Adsorption. So it's the oldest method of enzyme immobilization. What you basically have is a support, and the enzyme particles come and attach themselves to the surface of the support. So there's also an example about how Nelson and Griffin used charcoal to adsorb invertase. Coming to that point, does anyone know the difference between adsorption and absorption? I'm sure a lot of you have done CLP labs, so I think you might have encountered
Yeah,
this.
absorption is a bulk phenomenon, whereas absorption is a surface phenomenon.
Perfect. Yes, that's exactly it. So when you have absorption, you can think of it as a sponge where the water goes into the spaces or the cavity of this material. But when you have adsorption, like for example, charcoal or something, it's just a surface phenomena, and it like attaches itself to the surface. Perfect. That was just a side question. Moving on. So enzymes are adsorbed to the external surface of the support, and the support may be a mineral support, an organic support, or any modified cephalose and ion exchange resin. So these are just examples of what the support could be. The important thing is that you understand what is happening over here. There's a support, sort of like a backbone, and a lot of enzyme particles come, and they attach themselves to the surface of the support. Uh, we're going to explore this a bit more in depth. So there are generally weak bonds that are involved in adsorption. They're ionic interaction bonds, they're hydrogen bonds, and van der Waals forces. There's no chemical change as such. Adsorption is mostly a physical phenomena. So there's not much change in the chemical properties of the enzyme. So the weak bonds, because of that, they stabilize the enzyme to the carrier. They don't make it lose any of its properties. And over here, it says that the carrier particle size must be small. The particle size used can be from 500 angstroms to 1 millimeter. That's a pretty big range. And there's no pore diffusion limitations, because obviously it's a surface phenomena. So the enzyme is attached to the outside of a surface. It's not like there's a mesh of interconnected cross-linked polymers that make it difficult sterically for the enzyme to interact with the substrate. It's a surface phenomena. So the enzyme is attached to the outside of a backbone and it's free to interact with a lot of substrate molecules. Then this slide goes through the different methods of adsorption. There's a static process where you just allow them to come in contact with each other. You don't involve any stirring. Then there's the dynamic batch process. And there's the reactor loading process. You can go through these yourself. They're not very difficult. And they're not really involved with the understanding of what immobilization is. So I'd suggest you go through this slide yourself.
One last question. Like you said that these are not involved in immobilization. So do we need to write these in summary? Like if they are not involved in immobilization, then there is no point to write them in the summary.
No, I mean, they're not involved in your understanding of the immobilization process.
Yes. So if they're not involved in understanding, so what is the point, like in the writing these in the summary?
It's completely your choice, Achintya. Overall, the summary is up to you on how you want to write it. And as long as it demonstrates a good understanding of what immobilization is and what this PPT contains, till then it's fine. However, okay. Moving on to what the advantages of adsorption are. It's a relatively easy process to carry out. You don't need any reagents. Minimum activation steps are involved in this. It's comparatively cheaper and it's less disruptive to the protein than chemical methods. This is the most important point, I would say. Some of the disadvantages are basically the desorption of enzymes from the carrier can occur. There's always that disadvantage that you're trying to keep it bound to a physical entity, but for some reason or the other, it gets unattached. That is one of the disadvantages. You can write that disadvantage in any of the immobilization techniques. And the second is that the efficiency is less. Moving on to the covalent bonding technique. Over here, what you have is you have a distinct support on the side, and the enzymes are bound to the support via covalent bonds. So there's a distinct chemical change that's happening in the enzyme. It involves the formation of covalent bond between the enzyme and support. It's a widely used method, and chemical groups in the enzymes that form covalent bonds with the support are. This is a list of the various groups in the enzymes which can attach to the support. You have amino groups, there's hydroxyl, there's carboxyl, there's a whole bunch of them. So you can go through these if you want. Now, these are some of the important functional groups of the enzyme that provide chemical groups to form the covalent bonds. Now, again, you can go through this list yourself. There's 9 points. There's not much to understand over here. It's just a bunch of chemical groups that form the covalent bonds. Now, these are some of the carriers or the supports you use for covalent bonding. These are important. Certain carbohydrates that you can use, for example, as the support. You can use cellulose as a support. You can use agarose as a support. Does anyone remember where agarose is also used? Another application of agarose?
Agrogel electrophoresis.
Yes, perfect. It's used in electrophoresis, and you can also use it as a support for immobilization. Then you have synthetic agents like polyacrylamide, you have protein carriers, you have amino group bearing carriers. There are certain porous carriers like glass or silica. All of these can be used as the backbone or the support. and the enzymes will come and attach themselves to this. And the last point is that hydroxyl and amino groups, they form covalent bonds more easily. Now, this slide goes through some of the methods of covalent bonding. Um, it, it it addresses some of the finer aspects of what sort of reactions take place between the enzyme and the support. So there's bonding between the amino group of support and the tyrosyl or histidyl group of the enzyme. You can give these a glance yourself. It basically describes the chemical changes. I would say it's not very important. Now coming to the advantages of covalent bonding. Uh, this is one of the main advantages, the strong linkage of the enzyme to the support. Because the primary aspect is that you want the enzyme to bind to a support and be reusable. So if you have a strong linkage, that's definitely a plus point. There's no leakage or desorption problem because covalent bonds are relatively strong. It's a simple method. There are a variety of supports available, as we just saw in one of the previous slides, and there's also wide applicability. What are the disadvantages? This is the main disadvantage, the fact that a chemical modification of the enzyme, it might lead to functional conformation loss. You might lose the enzyme active binding sites where you have substrate molecules come and attach themselves. Enzyme inactivation by changes in the conformation, but it also suggests a temporary fix. This can be overcome through immobilization in the presence of enzyme substrates or a competitive inhibitor. Now, moving on to entrapment. This is the third process of immobilization. Over here, the enzymes that you have, they're physically entrapped inside a matrix. The bonds may be covalent or non-covalent, and the matrix used will be water soluble. So you have a matrix that looks like this, the sort of a mesh, and there are holes in the middle where the enzymes are held. And since it's water soluble, you put it into the substrate reaction mix, and the substrate particles can come and interact with the enzyme, and the reaction can be complete. So some examples of the matrices that you have, you can use polyacrylamide gels, you can use cellulose triacetate, you can use agar, there's gelatin. There's a lot of examples of what you can use as a matrix. Now, the form and nature of the matrix varies. The pore size of the matrix is adjusted to prevent loss of enzyme. The possibility of leakage of low molecular weight enzymes always exists. Agar and carrageenan, they have larger pore sizes. And yes, you can always adjust the pore size with the concentration of the polymer. You add more polymer, there's going to be more cross-linking. The pore sizes are going to go down. Entrapment of the enzyme can be used for sensing application. This point I'm going to have to come back to at a later time. We'll keep this to the side for now. There's not much success in industrial processes, and it's easy to practice at a small scale. Now, before we move further, does anyone have any questions?
Yeah, can you go back to slide 21?
Slide number 21.
Yeah,
Moment.
disadvantages of covalent bonding.
Yes. If it's
Yeah,
the fix, I'm going to have to come back to this as well because this is not something I understand very well either. But
okay, okay. Yeah, the last point.
yeah, I'll get back to that
Okay,
in the next practical.
sure.
Is there anything else? Okay, before we move on then, I'll pose a general question to the entire class right now. So over here in the entrapment step, one point is that the pore size has to be adjusted to prevent loss of enzyme, right? However, if you increase the concentration of the polymers, you can make the pore sizes very small as well. So can anyone see a sort of trade-off over here where you need to keep the pore sizes small but not too small, and why that trade-off exists? Can anyone explain that once?
Sir, because the enzyme is a macromolecules, and whereas the substrate is like glucose and fructose, so they are smaller molecules. So if we
Right.
increase the concentration of polymer, we want the pore size sufficiently small that the macromolecule doesn't pass through it, but whereas the substrate can easily go through it.
Right. You kind of have the right idea. So the basic idea is that if you add too much polymer, then your enzyme binding will be very tight. There'll be very little loss of the enzyme molecules. However, if the cross-linking is too rigid, then it's possible that the substrate and the enzyme might not even be able to interact with each other, because there's such a fine mesh of cross-linked polymers that um, the enzyme is held tightly, but it's held so tightly that it's not able to interact with the substrate. And you obviously do not want that. But you don't also want to keep the cross-links so weak that the enzyme starts escaping from the matrix. So there's somewhat of a trade-off there, and you have to uh, factor in both the points. Um, is that did does everyone understand that?
Yeah, makes sense. Yeah.
Yeah. Okay. Moving on. Yes. So, what are the different methods of entrapment? You could entrap it into a gel, of course. You could entrap it into fibers, or you could entrap it into microcapsules. Microcapsules are small entities that are formed by monomer mixtures like polyamine or calcium alginate. Moving on to the advantages and disadvantages. The advantages are that it's fast, it's cheap, there's a low cost matrix available. You need relatively mild conditions, and there's less chance of conformational changes in the enzymes, like we saw in covalent bonding. वहाँ पे we had the chances of conformational changes to be high, so that was a disadvantage. But when you're entrapping, if I mean you use non-covalent bond structures, then you have less chances of conformational change. The disadvantages are obviously the enzyme could leak out, the pore diffusion limitation, which I just talked about how if the pores are too small, then the substrate might not be able to interact with the enzyme. And finally, there's a chance of microbial contamination. Moving on to cross-linking or copolymerization. So over here, what we do is we don't really have a support or a matrix in the back. We just form covalent bonds between a number of enzymes and we hold them together. No matrix or support is involved in this. So some of the commonly used reagents are glutaraldehyde, there's diazonium salt. The technique is cheap and simple, but it's not often used with pure proteins. Can anyone guess why? Matlab, why would it not be used with pure proteins? It might have some.
Wouldn't it start aggregating?
Yes, there's always that possibility, but there's also the idea of covalent bonding. I mean, if you have a protein and it's in its purest state and it can catalyze a reaction very well, you wouldn't want to ruin that by sort of bringing in chemical changes in the structure. Does that make sense?
Yeah, got it.
Yeah. And apart from that, it is widely used in commercial preparations. The disadvantage is that polyfunctional reagents can sometimes denature the enzyme. So the polyfunctional reagents that we're adding for cross-linking of these enzymes, sometimes what happens is they interact with the enzyme directly and they denature them, which is obviously not what we want. Moving on, this is the final process. This is called encapsulation. So it's pretty self-explanatory. You have a semi-permeable membrane, and inside that membrane, you have a bunch of enzyme molecules. It basically involves making a capsule out of nitrocellulose or nylon. These are semi-permeable substances, and the effectiveness depends on the stability of enzymes, as it does in any process. That's fine. So the advantages are that it's a cheap and simple method, and you can immobilize large quantities of this enzyme via encapsulation. The disadvantage is, again, the pore size limitation. The semi-permeable membrane, if it has pores that are too small or too big, uh, interaction might not be able to occur. And only small substrate molecules are able to cross the membrane. They're basically the same point. So yeah, moving on. Now, so far what we've done is it has been the immobilization of enzymes. We're now going to move on to the immobilization of entire cells. Before we do that, does anyone have any questions? We're on slide number 30. It goes up to 35, so we're almost at the end of it. So if you have any questions, now is the time. Okay, I'll assume there are no questions right now. So far we've been immobilizing enzymes. Now this is a process where we're going to be immobilizing entire cells. So it's an alternative to enzyme immobilization. So basically what happens is you have certain cells, you have certain cells jahan pe, if you genetically modify them, they sort of become factories for producing proteins that you want. I'm sure you've done some of this in your molecular biology course where you might have uh, inserted plasmids into E. coli cells or things like that. So basically what happens is you have a microbial cells, you've genetically modified it so that it now becomes a factory for producing a certain enzyme. Now, instead of removing and purifying that enzyme from that mixture, from your culture, what you're actually going to do is you're just going to be immobilizing the entire cells inside some structure. So once you've immobilized the cell, it is constantly going to be producing those enzymes, and you'll have achieved uh, the same target which you were hoping to achieve with immobilization of enzymes. So it's a pretty effective method when the individual enzymes become inactive during immobilization. There's always that risk. The isolation and purification of enzymes is not cost effective. Like I said, you can, instead of isolating and purifying the enzyme from the culture mixture that you have, what you're doing is you're directly immobilizing the entire cell. So that might be more cost effective than this. Here, the enzymes will be active and stable for a long period of time. And the methods of cell immobilization are the same as we described for enzyme immobilization. There were 5 processes, as far as I remember. There was adsorption, there was chemical bonding, there was covalent bonding, there was a matrix formation, there were you had cross-linking of all the enzymes, and you had an encapsulation. So all of those processes can be used for cell immobilization as well. Now, what are the advantages of whole cell immobilization? Multiple enzymes can be introduced to a single step, because obviously if you have a cell, it's not only going to be producing one enzyme. You can modify it so that it produces multiple enzymes. So if you immobilize the cell, you're basically immobilizing 3 or 4 enzymes at the same time. Then the extraction and purification of enzymes are not required. The enzymes are stable for a long time. The native conformation of enzymes is best maintained, because over here you're not making any changes to the structure of the enzyme. The cell organelles like the mitochondria and chloroplasts, they can also be immobilized instead of the entire cell. So that's another branch that's another application that might branch out of this one. What are the disadvantages? The disadvantages are that the concentration of enzymes will be less compared to if you had just done enzyme immobilization. There could be a production of unwanted enzymes and unwanted products, definitely. And modification of end products by other enzymes could also take place. So these are some of the disadvantages. These are some of the methods. Like I said, we've already discussed all of them. They're very similar to enzyme immobilization. This is a table in which you have the methods of whole cell immobilization, the support material that is being used, the cells which we are immobilizing, and the reaction which gets catalyzed by these immobilized whole cells. So you can go through these once. You can find out which reactions are being catalyzed by which cells. It's not necessary to memorize this, obviously. You can just go through it once for your own interest. And that is it. That's the end of the presentation. Do any of you have any questions at this point? Okay, I assume everyone understood what was said. So, like I said at the beginning, there's going to be a report that you have to submit at the end of, uh, I mean, you're going to have to submit a report for this practical. So there are going to be 3 sets of questions, Usma. The first question is basically, I think someone should write this down somewhere. The first question is going to be to write a summary of this entire enzyme immobilization PPT that I just went through. I'll go through the important points, sort of the points that require understanding, not the ones that involve rote learning or anything. Try to limit it to, I would say, 350 to 400 words. I think that should be enough. I mean, there are 35 slides, so I think 400 words is a decent word limit. And now we're going to move on to the second aspect. This is the experiment that you guys would have done if you were on campus and under normal circumstances. So I would like all of you to, I'm going to have these 2 PPTs uploaded. So basically we have one PPT, which we just went through. I explained the basics of enzyme immobilization to you. Apart from that, there are 2 PDFs that I would like you to go through. The first PDF is basically the experiment. So in short, I'll tell you what is going to happen. You would have done 3 methods of enzyme immobilization by gel entrapment, and you would have compared the efficiency of the 3 different types. And you'll see that at the end of this, there's a group of 8 questions. Can you see this, everyone? Just one person just say yes or no.
Yeah.
Yeah. So
Yes.
after you go through the procedure and everything, the discussion and all of that, I'll need you to answer these 8 questions in your report. Keep it short. They're not very difficult questions. Now, some of these questions are basically based on the experiment that you guys have not done, right? So you can either, for example, in those questions, let me see. Right, like question number 5, was there any shift in the optimal pH or temperature due to immobilization? Did you expect any? Now, this question, obviously, if you haven't done the experiment, it cannot be expected that you'll give the answer to this. but maybe you can just give it your best guess. You can just write down what you think will happen, and we'll grade you on that as well. As long as you've given proper reasoning, there's no reason for you to not get marks. So अच्छे से देना ज़्यादा, you don't have to think about it too much. The theoretical questions, answer them as well as you can, and the practical questions which involve the experiment, write down what you think will happen, try to give a reason or two, and we'll check that. All right, and then there's another PDF. This is a review paper on the overview of immobilization techniques. You can go through this if you want as well. This is basically for extra reading. It's not a very long paper. It's, I don't know, I would say it's like, it says 17 pages, but then there's a bunch of references at the end which don't really count. So close to, I would say, 12 pages. You can go through it, and I would like you all to write down the questions that you're going to be submitting in the report. So, like I said, there's going to be 3 sets of questions. The first question is that you're going to have to summarize the slides. Try to keep it between 350 to 400 words. Try not to exceed it. If it's less than that, that works. Just try to capture all the main points of the PPT. That's the first question. The second is the set of questions at the end of this enzyme immobilization procedure, the experiment that you were going to do. And the third question is based on this paper that you're going to read. This is for further reading. So I would suggest that you note this question down. Which techniques would you use if your enzymes turned out to be inactive after immobilization? And how would you identify that they were inactive? All right, is anyone noting this down right now? Because I'm assuming that this meeting is being recorded. Yes, it's still being recorded. So you can write down the question right now, or you can come back to this lecture later on and write it down somewhere. But the question is, I'm going to repeat it once. What techniques would you use if your enzymes were inactive after immobilizing? What would you do to identify that they were inactive in the first place? All right, did everyone hear that? Can someone confirm?
Yeah, noted.
Yeah, that's perfect. So I'll tell Professor Mukherjee to upload these 3 files, the single PPT and the 2 PDFs on the BBL433 group, and you guys can take it from there. And as far as the deadline for this report is concerned, I think that's something Professor Mukherjee will tell you. And in the meantime, if any of you want to contact me, I'll just a moment. Is there a I'm not on the group, so I can't really send a message right now. But isn't there supposed to be a blackboard option as well, a whiteboard option as well? Does anyone know how that works? I'll stop sharing my screen.
Ashutosh, I guess you can drop a message in the chat and then like people can contact you via Teams.
No, I'm not in the a part of the team right now, so I can't really drop a chat. It says chat in the channel meetings are only available to team members. I haven't been added yet, so we'll have to figure something out. Acha, okay, you guys can write down my entry number, and I think webmail ke through you can contact me after that. So my entry number is 2017 BB 50060. I think I had one course with Samuel once, I think. So I think the only difference in our entry numbers is the 2018 and the 2017. The last digits 50060 are the same as far as I remember. But ठीक है, मतलब I'm sure you can convert my entry number into my Kerberos ID and contact me via webmail if any of you need help. So, yeah, that's all. Why don't you guys stay on for like 2 minutes more? I'll just confirm with Professor Mukherjee that there's nothing else he wants to share with you guys. But from my side, that's all. Just give me 2 minutes and then I'll give you the go ahead to leave the meeting. All right? Okay, everyone. I just spoke to Professor Mukherjee and he says that there's nothing else to discuss. So that's okay. All of you can leave now. And yeah, thank you for attending.
Thanks,
Thank
Siddesh.
Yeah.